ADView is a tool to help biologists to compare one phylogenetic tree against a collection of trees. On the left is a reference tree with 103 species of plants shown as a dendrogram in full detail. Say we want to explore a group of algae highlighted in blue, and also the bryophytes in orange, the vascular plants in green. On the right is a tree collection with 68 trees of the same set of plants shown in multiple levels of detail. Each of these aggregated dendrograms in the middle represents one tree, where the 3 selected subtrees are highlighted, as well as their topological relationships. If we hover over a particular block, there will be black dots in the reference tree, showing the species in the block. This one has all the blue species, but this one is missing one called Spirogyra. We also group the individual aggregated dendrograms into clusters based on their topology, resulting in only 2 possible relationships between them. These 2 clusters represent 2 different hypotheses, whether bryophyte and vascular plants are direct siblings or have other species between them. However, most of the trees, precisely 64 out of 68, agree that they are direct siblings. Let's turn to the tree distribution view. Each row tells us about the agreement and conflicting alternatives with respect to each selected subtree. For example, the second row for the bryophytes has 3 segments. The first segment agrees with the reference tree, as you can see from the black dots in the orange group of the reference tree. This second subset of trees is missing the top subtree, the group of homeworts, and this third small subset of trees also excludes the middle liverworts group. If we want to compare multiple segments simultaneously, we can persist the black dots in aligned columns. When we hover on a subset of trees, they are also cross-highlighted in other rows in the tree distribution, as well as the aggregated dendrograms. We can select one tree and pairwise compare with the reference tree in full detail. We can easily spot how the 2 trees differ in the highlighted groups. We can also highlight up to 5 groups between the 2 trees. Let's take a look at a larger dataset that compares a species tree and 424 gene trees. The top histogram here shows the distribution of important branch attributes. We can highlight the branches that have low agreement, that is, high conflict, in the tree collection. We select the land plant subtree in blue. Here we see a long tail effect in the tree distribution, indicating there are a lot of trees that do not agree with the reference tree nor each other. The dotted block at the bottom of the aggregated dendrograms represents missing species, which are extremely common in gene trees. Let's see why this tree conflicts with the reference tree. The messy case here, with many small separate blue blocks on the right, is probably caused by incorrect rooting. The outgroup species on top on the left that are at the root are all in the missing group at the bottom on the right in this gene tree. A different tree has a large continuous blue block, showing it agrees with the reference tree despite the missing species. We can click on other subtrees to explore their topological relationships. In the pairwise view, we can see when colored blocks are contiguous or spread out on the other side. We can also see how the blocks connect to each other in the clustered aggregated dendrograms. Here we see 15 clusters, representing many more hypotheses than in the previous dataset. It's typical to have many conflicts between a species tree and its associated gene trees.